Hello students, today again we are going to continue with biology chapter number 4, that is molecular basis of inheritance. In this topic there are two parts. We have completed in our last lecture the first part of the chapter, that is gene, its nature, expression and regulation. Today we are going to start with second part of the chapter, that is genomics. Okay, we have completed the first part of the chapter. We have studied everything about gene, its nature, expression and regulation. So we are going to start with the second part of the chapter. Okay, in our last lecture we have covered the topic lac operon and role of lactose. Okay, in detail we have studied about the lac operon. We have studied the structure of lac operon and function of lactose. Today we are going to start with second part of the chapter, that is genomics. When I say genomics, we are going to study about genomics in very much detail. Before starting with the topic genomics, let us know what do you mean by genome? What is the meaning of genome? When I say genome, this term genome was introduced by H. Winkler. Okay, it was introduced by H. Winkler in 1920. You need to remember this question for objective for your examination. Okay, in exam purpose point of view. So this term genome was discovered by H. Winkler in 1920. Okay, the term genome denotes the complete set of chromosomal and extra chromosomal genes present in an organism, including a virus. Means we can say that it is the total genetic constitution of an organism. Okay, we can say that it is the total genetic constitution of an organism. Okay, let it be any organism. The entire genetic constitution of an organism is known as genome. We can also say it is a complete copy of genetic information, that is DNA. Okay, we can say it is a complete copy of genetic information in cell. Okay, that is DNA. Genetic information means it is a DNA, complete copy of genetic information. Even we can define it as one complete set of chromosome, okay, of an organism. As I have told you, one complete set, you can see here, complete set of chromosome in an organism. Okay, so there are three definitions. You can go with any of the definition. First one, it is the total genetic constitution of an organism. Total genetic constitution. Okay. Second, it is the complete copy of genetic information. That means it is related to DNA. Complete copy of genetic information means it is a copy of DNA. Then the third definition, we can say it is the one complete set of chromosome. When I say chromosome, it can be monoploid or haploid. Okay, it can be present in a single form or it can be present in a double form, that is monoploid or haploid of an organism. Okay, now moving on to the actual topic, that is genomics. Okay, when I say genomics, okay, we will study about its discovery, introduction part. We will study the type of genomics. We will study the application of genomics in detail. Okay, starting with genomics. What is genomics? Okay, when I say genomics, what do you mean by genomics? Genomics is an area within genetics. Okay, it is an area in genetics that concerns the sequencing and analysis of organisms' genome. Okay, organisms genome we have already studied. What is genome? Okay, so it is the concerned with the sequencing and analysis of organisms' genome. Then it involves the study of all genes at the DNA, mRNA and proteome level as well as at the cellular or tissue level. Means when I say genomics, it is concerned with the study of all the genes. Okay, every gene in DNA, mRNA, proteome, level as well as cellular and tissue level. Every gene is concerned here. Okay. The term genomics was first coined in 1986 by T.H. Roderick. That means Tom Roderick. Okay. It was introduced by Tom Roderick. He is a genetist at the Jackson Laboratory in Maine during a meeting about the mapping of human genome. In simple terms, in easiest words, I can define genomics as the subdiscipline of genetics devoted to the mapping, sequencing, and functional analysis of genomics. Okay, that means we will first do the mapping, we will know the information, then we will sequence it, and then function analysis of genomics we will do. Okay, it is the study of genomes through analysis, sequencing, and mapping of genes along with the study of their functions. Okay, now when I say introduction, initially how this genomics was studied, comparative study was done. Okay, what was done? The sequencing of yeast, Drosophila, and mouse was done in order to facilitate comparative studies between human and other organisms, which are commonly used for genetic studies in laboratory. Okay, what was done initially? The sequencing of yeast, Drosophila, and mouse was done in order to facilitate the comparative studies between humans and other organisms that are commonly used for genetic studies in laboratory. Now, several microbes like bee, tomato, and other crop. Okay, microbes, bee, tomato, and other crops are now actively sequenced. Okay, are now either actively sequenced or considered for sequencing. Initially, yeast, Drosophila, mouse was used for sequencing, and this was done in laboratory. Now, what I used? Microbes, bee, tomato, and other crops are actively used, and they are considered for sequencing. Okay. Now, let us move on to the types of genomics. I can say types, or I can say genomic study is classified as structural genomics and functional genomics. Okay, genomic study is classified as structural genomics and functional genomics. Say even there are some other classification, but as per your textbook, only you have to study these two types only, that is structural genomics and functional genomics. So let us study structural genomics. Structural genomics involves mapping, sequencing, and analysis of genome. It is only concerned with mapping, sequencing, and analysis of genome. Whereas functional genomics deals with study of functions of all gene sequence and their expression in organism. Okay, I repeat, functional genomics deals with the study of functions. It is, the name itself indicates functional genomics means it is related to functions. Okay, it deals with the study of functions of gene sequences and their expression in organism. Okay, now moving on to very important part of this topic, that is application of genomics. When I say application of genomics, this is very much expected question in exams. Okay, application of genomics. So one by one we are going to study all the applications. Okay, structural and functional genomics is used for different purpose. Both these applications that I am going to discuss, these are application for applied for both structural and functional genomics. We have studied about structural and functional genomics. Now, how they are applied? They are useful for different purpose. First one, it is used in improvement of crop plant. Okay, genomics is used in crop improvement and human health and livestock. Okay, it is used in improvement of crop plant, human health and livestock. How? It is useful and provides information about genome size, gene number, gene mapping, gene sequencing, evolution of crop plant, gene cloning, then identification of DNA markers, marker assisted selection, transgenic breeding, construction, then QTL mapping. You all did not study all these points in detail. Just remember this point that genomics is useful in crop improvement, okay, human health and livestock. Moving on to the second application, that is, it is useful in different sectors. Okay, genomics research can be applied in number of different sectors, including medicine, biotechnology and social science. Okay, the genomic research can be applied in number of different sectors like medicine, biotechnology and social science. It is applied in the medical field, biotechnological field and social science. Okay, now moving on to the third application of genomics, that is, it helps in the treatment of genetic disorders through gene therapy. Okay, it helps in the treatment of genetic disorders. All the genetic disorders can be treated with the help of gene therapy. By the help of genomics, gene therapy, we can treat the different types of genetic disorders. Next application is, it is helpful in agriculture. Okay, it is helpful in agriculture. Genomics has also helped in agricultural field. It helps in the improvement in the yield and nutritional quality of rice and various other crops also. It helps in the improvement in the yield and nutritional quality of rice and various other crops. Okay, it is used in agriculture to develop transgenic crops having more desirable characters. We can say that doubling of world grain production in last 50 years. Almost the production has been doubled with the help of genomics. Okay, moving on to the next application, forensic analysis. Okay, genetic markers developed in genomics have applications in forensic analysis. Okay, genetic markers have developed in genomics have applications in forensic analysis. We can say that information and clues obtained from DNA samples found at crime sites have been used as evidence in court cases, and genetic markers have been used in forensic analysis. Okay, so this is the major application and the very important application that it is used in forensic analysis. Okay, next we can say that it can lead to introduce new gene in microbes to produce enzymes, therapeutic proteins and even biofuels. Okay, it can lead to introduce new genes in microbes to produce enzymes, therapeutic proteins and even biofuels. Application part is very much important. I will repeat the applications fast in short. Okay, just remember, jot down the points for application that you have to remember in exam. It is first point, it is useful in crop improvement. Okay, it is useful in crop improvement, human health and livestock. Second application is, it is used in different sectors like medicine, biotechnology and social science. Okay, third application is, it is useful in treatment of genetic disorders through gene therapy. Okay, gene therapy. Fourth application is, it is helpful in agriculture. Okay, it is helpful in agriculture to develop transgenic crops and we can get the desirable characters of the crops. Next application is, it is useful in forensic analysis. Okay, and the last application is that it is useful in microbes to produce enzymes, therapeutic proteins and even biofuels. Okay, thank you.